Now that you're familiar with DNA structure and function, we're going to talk about how DNA replicates or gets synthesized. The whole reason behind this is if you're making a new cell because your body's just doing typical growth, or if it's repairing things, if an old cell died, or of course, babies are having to make lots of new cells to get their bodies going. So the whole idea is we want to have the exact same DNA as the original cell. That is the point, okay? So in order to do that, we're going to need to look at our base pairs, and we're going to want to replicate that, okay? But notice how the base pairs are kind of inside the double helix. So basically, here's DNA replication in a nutshell. You got to open up this double strand so you can get to the inside. You got to figure out the matching base pairs and then put it back together again. That's it. But as it turns out, that's it. It's actually kind of complicated, and it takes lots of different molecules and enzymes and proteins to make this happen. Here's a list of some of the proteins that are involved. You can, if you want to right now, pause the video and write down each one individually, or you can just wait and write them down as I mention them, whatever you prefer, okay? So let's get into how DNA replication actually happens. So remember, we got to separate that double helix to get started. So there it is, initiation. Here's how we get started. There's an enzyme called DNA helicase, and this is going to come in and break apart the double helix. It's going to break apart these hydrogen bonds in between our nitrogen bases, okay? Now, if you can imagine, DNA is it's a double helix, right? It's coiled. And so imagine taking a slinky and trying to break it apart and uncoil it. Ooh, that slinky might snap or break. So there is another enzyme called topoisomerase, okay? And topoisomerase is going to come just ahead of the DNA helicase. So it'd be just over here, right in this picture, okay? And what it's going to do is it's going to basically help ease some of the stress that the untwisting is causing, and it's going to keep these from snapping into pieces because we wouldn't want that to happen, okay? So topoisomerase just kind of helps ease that uncurling motion, okay? Helps take out some of that extra stress. So, okay, we've got our topoisomerase helping ease that strain. We got helicase now that's opening this section right here. But what keeps it from closing back up? These guys, single-stranded binding proteins, or SSBPs, okay? And so they keep them open. Cool. So now we've got an open segment of DNA. Let's start actually making a new piece of DNA. Well, before you get started, as it turns out, all DNA replication starts with a little bit of RNA. What? Yes. Okay. So as it turns out, in order for DNA to get made, it needs a little piece of RNA to grab onto. It's kind of like, on your coat at the bottom, you got the zipper, and there's that little flat tab that you got to have to get your zippy part onto the zipper. Okay, your RNA primer is like that flat tab. Okay, you have to have it in order to get it going. So there's going to be an enzyme called primase that comes, and it's going to put down a small little segment of RNA, 5 to 10 base pairs in length. And so that's going to be where things are going to be starting right there. Okay, and so now an enzyme called DNA polymerase, because it's making a polymer of DNA, but specifically DNA polymerase 3. There are many different types of polymerase. So DNA polymerase 3 comes in, finds that RNA primer, okay, and is able to latch on now thanks to RNA primer, and it is going to start at this 3' end here, and it's going to move towards the 5' end, okay? And so it's going to look at this very first base pair. And if it's an A, it's going to find a T because there's all of these nucleic acids floating right here. It's going to find a T, boom, that's a T. If this second one right here is a G, then it's going to find a C, put that one right there, and so on. So it's going to keep reading each one of the parent strands and is going to find the complementary base pair and start adding it on. And this thing is just going to chug all the way down the length of this piece of DNA. Okay, it's going to go from 3' to 5'. Okay. And then you can imagine it's going to at some point get to the end. Cool. Are we done? Well, not quite, because is RNA present in our DNA strands? No, it's not. Okay, so there's going to need to be another enzyme, DNA polymerase 1, that comes back in. It's going to take out this RNA primer and put in a couple pieces of DNA. But here's the thing. These new pieces of DNA are not connected to the pieces of DNA that was made before. And so now we need a final enzyme called ligase that's going to join those two pieces together. So now we have one continuous piece of newly made DNA. Okay. Yay, we're done. Hooray. This one got copied all the way. We replaced our RNA. Everything's done, right? Nope, not exactly. Okay. Because notice that this is happening on this strand. But what about this strand? Is the process happening on this strand too? Yes. Yes, it is. Okay. So it's going to be happening on both strands, but it's going to be happening in opposite directions, if that makes sense, because DNA polymerase moves from 3 to 5. And so down here, 3 to 5 is going this way. Whoa. Okay, so now we're going to get into exactly how it happens on the other strand. Okay, so because of the fact that we've got kind of it happening in both strands, you can almost think of this as kind of like a little bubble here, okay? And so as it turns out, when there's DNA replication, you have what are called replication bubbles. In prokaryotes, because the chromosome is a circular chromosome, this bubble just continually grows until you have 2 circular pieces of DNA. In eukaryotes, however, we have linear chromosomes, and so we're going to have one helicase going this way, one helicase going this way. But to save time, we're also going to have some happening here and here and here and here. And it's going to keep going in both directions until the helicases all meet up with each other. And these bubbles get bigger and bigger until it all joins into 2 big DNA molecules. Okay. So again, when we're doing replication, because of the fact that we're going to have a helicase going 3 to 5 one way and 3 to 5 another way, we're going to have 2 helicases actually. All right, so here's my DNA, 3 to 5, okay? So this helicase is going to the left and is opening things up to the left. This helicase is going from 3 to 5, so this one is opening up things to the right. So we got 2 helicases going in opposite directions. That means we're also going to have 2 topoisomerases going in opposite directions, one going this way to help ease the strain over here, and one going this way to help ease the strain over here. You with me so far? Okay. Here's the thing, though. As it turns out, we're going to have this polymerase going this way, and we're going to have a polymerase going in the other way. Now, this sounds really easy, but this is actually going to lead to some complications, okay? And this is going to end up creating what we call a leading strand and a lagging strand, okay? And so here's the leading strand is our helicase is going this way, going to the left. And so our polymerase is going to follow right behind helicase. It's just going to keep motoring until it hits the end or the next bubble. Great. So it's going to be able to build one long, huge one. But this strand is going in the opposite direction. Oops. And so it's not going to be able to build one long, huge one. Okay. And we're going to get into that the why for that in a second. But this strand is going to take a little bit longer. It's not going to be able to do it in one. And so this strand is called the lagging strand. Okay. So imagine for a second right now that I want you to like cover up from here on. Imagine that helicase is right here. Okay. And nothing has been opened to the left. So imagine that we got our RNA primer, we have our DNA polymerase 1, it's coming in, it is laying down new pieces of DNA. But on this lagging strand, now we're going to uncover this imaginary section. Now helicase has gone to the left. More of this is open that wasn't open before. Our DNA polymerase can only move to the right. So I'm going to need a new piece of primer, RNA primer put on here. Another DNA polymerase 3 is going to come on in and build new DNAs until it hits this RNA primer. And it's going to pop off. And then guess what? Helicase has busted up some more, and so now there's more over here that hasn't been done. So here's another piece of RNA primer. A new DNA polymerase 3 is going to come in and play down new nucleotides till it hits this old primer. Okay? And then, of course, We're going to have DNA polymerase 1 come in and get rid of that primer. We'll have ligase come and join the old and the new. But you see how we're kind of like laying this one down in short segments, whereas this one is just going to be able to be in one continuous line. So this one continuous line is called the leading strand. This other one is called the lagging strand. And these little chunks down here are called Okazaki fragments. Okay. Here is just another picture of that showing an Okazaki fragment. Okay, here's the primer. Here's DNA polymerase 3. Okay. And so this right here is the same thing down here. But now our helicase has opened up more. There's topoisomerase. Here's our helicase. It's opened up more. These are SSBPs. Okay. So now all of this is available now. And so we have a new piece of RNA primer. Here's another DNA polymerase going till it hits this piece, and then it's going to stop. Okay. And now we'll have polymerase 1 come in and munch out the RNA primer, and then we'll have DNA ligase joining. But now look, here's even more that's available. So we're going to have another piece of primer and another piece of DNA made by the polymerase 3. So these are the Okazaki fragments. I know it's kind of confusing. We'll go over it in person. At the end of this process, okay, here we go. We open up our DNA. We build a new piece, some of it in fragments, but we're not going to get back into that. And at the end, we have 2 new pieces of DNA. But here's what I want you to notice. Notice that this one has 1 template strand and 1 brand new strand. And this one has 1 template and 1 brand new. And the fact that you get 1 old and 1 new, this is called semi-conservative replication. You don't get both olds together and both news together. You get 1 old and 1 new. Okay? Pretty cool. So we get 2 identical molecules of DNA. It's identical-ish, okay? This is happening so fast. In bacteria, it's 500 nucleotides a second. In humans, it's 50 a second. And we do get errors. Originally, about 1 in 100,000 of our nucleotides are put in incorrectly. But different DNA polymerases come in and they're like making sure that A is with T and C is with G. And if for some reason an A is connected to a C, it's going to rip it out and put a T in its place. Okay. And after DNA polymerases come in and do the proofreading, instead of 1 in 100,000 base pairs, there's 1 error in every 10 billion, which is 1 out of every 3 cells. There's 1 teeny tiny itty bitty error. Imagine typing like 10 billion letters and only getting 1 wrong, right? So it's really good. Small errors, but really good. Okay. So that is how DNA replication occurs. I know it's a little bit complicated, but here are some of the main things to know about it. Please see your textbook if you need to. It goes through all this in depth with great pictures and huge paragraphs, so that can help you. And we will also be looking at this together in class as well.